Hi everyone, welcome back. Wow, we are getting close to the finish line here. So we're going to finish off topic 7 this week, but we also have our second lab report due today. So hopefully you've had a chance to work on it and are just finishing it up to submit it. So this enzyme lab report, this is topic 5, step 6. This is due today, so to help you if you haven't even started on it, do make sure you watch my instructor video. So the enzyme lab report instructor video, I walk you through every component of the lab report. So this is a good resource to have on hand when you're pulling everything together. So once you've completed your lab report, you'll go ahead and submit it via the enzyme lab report link. Do make sure that you grade your lab report against the rubric before you put the submit button, just to make sure that you haven't left anything important out. So I use that same rubric to grade your report. So if you forgot to include a title or you did not explain your hypotheses, I will just mark it in the rubric. So use that rubric to help you. So this is due by midnight tonight. Also, just like any other week, we have the new topics for the week, the due dates calendar. So let's go ahead and take a look at what we will be doing. So we're going to be working through the rest of the topic 7 assignments. So we've completed 4 of the 8 assignments already. So we're going to continue on through topic 7, step 5 through step 8. And then we will again be working through our second lab report and start on our third and final lab report. Okay, and then just a reminder, you should be continuing to work on your integration project. So this project, especially since you've gained some understanding of what genes are and how there's different alleles for a gene and how that can contribute to your genetic disease topic. You know, those are some of the information we've gained this last week, so you should be incorporating your new knowledge into your integration project. Okay, so let's take a look at our assignments this week. So we'll start with gel electrophoresis virtual assignment. So last week we talked about the bitter taste lab. So you had a chance in topic 6 to taste the PTC paper and determine if you're a strong, a weak, or a non-taster of PTC. So then last week you learned about how we can isolate that specific bitter taste gene. The techniques that are used. Okay, so that was in topic 7, step 3. So you learned about how the DNA was extracted and that particular gene was isolated and amplified using PCR, polymerase chain reaction. So this week, we're going to take that information from the PCR machine, which is basically just thousands of copies, or more than that, millions of copies of the bitter taste gene, and how we can decide if we are strong tasters, weak tasters, or non-tasters. Okay, so do we have 2 dominant genes? Alleles? Do we have a dominant and recessive allele, or are we homozygous recessive for the bitter taste gene? And so we do that using gel electrophoresis. So in gel electrophoresis, we do have this virtual lab that you will take a look at. Okay, so you'll press play and it'll go through the different parts. So this includes adding a restriction enzyme to our sample. If this was an in-person lab, we would actually add that. And then that restriction enzyme would cut only the genes that have the particular bitter taste to them. Okay, so the genes that are cut are the ones that are the bitter taste genes. So the non-tasters, if you have a non-taster gene, it doesn't get cut. And so what ends up happening is that in the sample, some students that are strong tasters, they're going to have a lot of small fragments of DNA. Whereas the samples of the students that are non-tasters, all of their fragments are long, okay, so because they were not cut. And then finally, those that are weak tasters, they will have some long and some shorter segments. And so what we do is we go ahead and run this through gel electrophoresis, which is just a machine that has an electrical current, and it allows the sample to move. Okay, so the smaller the cuts are, or the smaller the pieces are, the faster the DNA will travel. So you can imagine if you are a strong taster, most of your DNA will travel pretty far on the gel electrophoresis, whereas those that are non-tasters, they would not flow that far. So in this, once you kind of get an idea of what the experiment is like using the virtual lab, then you're going to take a look at the workbook. So on starting on page 265, there's nothing really there. But on 266 and 267, you're going to learn about where the restriction sequence is and how to cut it. So what are restriction enzymes? And so you're going to practice cutting the restriction sequence to create the RFLPs. So we have on page 267, you will actually cut out those DNA double helixes and cut them at the HEY3 enzymes, restriction enzyme site. Okay. So once you do that, and by the way, you basically, it's pretty easy. You're just looking for that restriction sequence. Okay, so if you see G, G, C, C on one side, C, C, G, G on the other side, you'll cut them in half. Okay, so then you're going to use that information that you just gained through the virtual lab, as well as cutting of your DNA, to answer the questions on page 269. Okay, so on 269, use the virtual lab to answer those 7 questions, and then on the next page, 271, you're going to use your samples that you cut and put them in the simulated gel electrophoresis on page 271. Okay, so you're going to see how basically far the samples traveled and then what the differences were. Okay, so once you have completed this assignment, then locate your particular assigned questions and post those no later than this Sunday, March 3rd. The next one is we're going to look at uses for DNA analysis. Okay, so this is going to be basically, how do we use these gel electrophoresis besides looking at determining if you have a, bitter taste gene or not. So a lot of it is used for like DNA analysis. Specifically, the examples we are going to be working through this week is how we can use it for paternity cases. So, who's the daddy of the, and then also for crimes, for forensic cases. So we'll look at both of these. So we have, for this example, we do have these auto radiographs that you will be using to help complete the page 275. But before you look at these, do make sure that you're looking at page 273 and learning how to read them. So if you take a look at number 1, you can see that we have these 4, basically they're auto radiographs. And at the top we have M, C, and F number 1 or F number 2 columns. And so the M stands for the mother, the C stands for the child, and then the F stands for the possible father. And so what you're going to want to do is you're going to want to see the bands that match the mom for the child and the bands that match the possible dad for the child. Okay, so the child's the one in the middle with all the bands represented. Remember, the child gets half of its DNA from mom and the other half came from dad. Okay, so you can see in the first example that the child got the first band from the mom, and then the second band came from the dad because they match up. Okay, so you'll use this to help you determine which father could be excluded. So could father number 1 or father number 2 be excluded? So based on which bands overlap. Okay, so if the child does not have any overlapping bands with the father or very few, then most likely that father is not the biological father. If there's multiple to many bands overlapping for the child, then we know that that's a pretty good indication that that child was fathered by that particular father. So you'll use that radiograph on page 275 to answer the first question. The second question, we're going to be looking at a forensic case. So in this case, it's unfortunately, sorry if this is triggering, is a rape. And so basically we have the victim, the evidence that was collected from the victim's vaginal canal, and then we have two suspects. So the suspect's DNA that was recovered. So basically we have to determine, was it the first suspect or the second suspect that raped the victim? And so on the figures on page 274, the first column would be the victim's DNA. The second is the evidence that's collected. So that has her DNA as well as the suspect's DNA. And then we have the suspect number 1 DNA on the right of the first box. And then the 2 boxes below we have the suspect 2's DNA. So you're going to basically look at where the suspect's DNA overlaps in the evidence. And if there is no overlap, then we can exclude that suspect from committing the crime. If there is overlap, then we can suspect that it was probably that suspect that committed the crime. So once you get an idea of how to do that forensic case, then we're going to actually apply it to some actual auto radiographs. So starting on page 275 and using the auto radiographs from this link, it'll help you determine, see, these are actual ones, so it helps you determine, in this case, this is a paternity case. So you can see the way that it's read, we have the mother, the child, and then the father, okay? So you can use, if you want, down here, the child/father, but I just look at basically the same 3 columns that we saw for the paternity case on page 273. So it's just on its side, right? So we have mom, child, and father. So where the child overlaps with mom, we know that, that child is obviously biologically linked to mom. But then we have to look at the other DNA of the child and see if it overlaps with dad. And if it does not, then we can exclude that father. Okay, so in this case, this first one, we see that the child's DNA does in fact overlap with mom, which makes sense. But then if we look at the father's DNA, none of it overlaps with the kid. So I would exclude this father as the possible father. Okay, then you can kind of look at the second one. So here's mom, here's child, here's father. Here's, again, third one, mom, child, father. So you're going to use these 5 different paternity cases to determine who was the alleged father and who is not. Okay, and then scrolling down, so that'll be answered on page 275. And then on page 275, we also have an example of a actual forensic case. So same thing, where we're looking at the victim's DNA, and we're going to compare that victim's DNA to the evidence that was collected at the crime scene. So in this case, we have the victim's DNA, we have 2 possible suspects. We have the female cells recovered as well as the sperm DNA that was recovered at the crime scene. So obviously the female cells were collected from the victim, but then we're trying to figure out whose sperm that was. Was it suspect 1 or suspect 2? And then you can see how, in this case, the husband's DNA was also collected and to be used as an exclusion. So you can figure out which suspect can be excused as the suspect, excluded, and which one probably committed the crime based on the evidence that was supplied. Okay. So again, once you've completed those radiographs, then you're just going to answer the questions on page 276 to determine the basically how accurate DNA evidence is. Okay, so when we do introduce DNA evidence into a paternity case or into a forensic case, you know, how accurate is it? Is this really strong evidence or is this weak evidence? So you'll be doing some calculations to determine the validity of the auto radiographs. So once you have answered all of the questions, you're going to post your answers to all of these pages. So just, you know, take a picture of each one of the pages starting on page 273 and then upload them and submit them. Okay, or you can just, if you don't want to do pictures, you can just answer the individual questions. Okay, so that is due again on Sunday. And then moving on to everybody's favorite, the blood typing. This one, you will be using your Eldon card, so you should have received that in your lab kit. And so this is a way that we can actually you can determine what your own blood type is. So the first thing that you're going to be doing is reading page 277 to 278 to learn about what the ABO blood typing system is all about. So, you know, what does it mean if you're type A or type B or type AB or type O? And what are your possible genotypes if you're type A or type O or etc. Okay. We're going to just, you know, we have some multiple alleles involved here. So use your understanding of codominance as well as incomplete dominance to be able to set up the Punnett square for this scenario on question number 3, where we have a heterozygous female that's blood type A and a male that's blood type O. So think about, if she's heterozygous, that means she has one dominant allele and one recessive allele. She's type A, what's the dominant allele? If she's heterozygous, she must have the recessive allele as well. So what would be the recessive allele in this case? Okay, so use that to help set up the Punnett square on page 277, and then figure out the possible genotypes and phenotypes of the possible children. Okay. Then you're going to actually test what your own blood type is. So we do have a video to walk you through how to complete the Eldon card blood typing. So you should have you know, everything present in that little kit, in your lab kit. When you do this experiment, it really helps if your hands are warm. I know sometimes my hands get really, really cold, and if I try to complete this test where I, you know, use the lancet to draw a blood sample from the tip of my finger, if my fingers are cold, it's not going to draw very much. Okay, so do try to make sure that your hands are warm. Sometimes just sitting on them for a couple of minutes or running them under warm water will help. And then when you're taking the blood sample with the lancet, choose your non-dominant hand excuse me, to take the sample from. And I usually like to do it on the side of my finger by my nail instead of on the pad itself, just because it's a little less cumbersome if you're going to be typing or texting or something like that afterwards. Once you've collected your blood sample and basically put it through the little Eldon, what is it, like a little card thing, then you're going to see where clumping occurs in your blood. So if there is clumping, then that means that you are present for that antibody, okay? So if you have clumping, that means that you are positive for that blood type, okay? So if you have clumping under anti-A, that means that you have the A blood type. If you have clumping under the B, that means you also have the B or the B blood type. If you have clumping under the D, that means that you're Rh positive. If you don't have any clumping, that means that you're type O. So once you figure out what your results are, make sure you take a picture first of all, before I forget. Take a picture of your Eldon card with your blood sample showing the clumping or non-clumping. And then you're going to use page 281 to determine what your blood type is. And then you're going to submit that picture of your Eldon card with your results as well as page 281. And you're going to submit it to the at the bottom of our assignment here. Okay, so it's just page 281 showing what your data results were from your Eldon card and what your blood type was, as well as the picture of your Eldon card with the blood included, okay? And then you can dispose of the card afterwards. And then the last assignment is the human pedigrees. So we've been talking about a lot of things with like Punnett squares and predicting outcomes of the possible children. But a pedigree is a little different because we can actually look at the history instead. So looking at your relatives, like your parents and your grandparents and your great-grandparents, and seeing where that particular trait that we're studying has come from, which side of the family and so forth. So you're going to be looking at page 282, and first of all, we're just studying the symbols used on pedigrees. So you'll notice right away that we have circles for females and squares for males. If it's shaded, if that circle or square is shaded, that means the person has whatever trait we're talking about. And then you can see how if a circle and a square have a horizontal line between them, that means that they are married or at least had sexual relations that produced a child. So if the vertical line comes off of that horizontal line, then you indicate the child. So if the child is a circle, that's a girl. If it's a square, it would be a boy. If there's multiple children from that union, so we might have circle, square, circle, or vice versa, that means those are siblings of that couple. Okay. And then you can see some of the other ones that are a little more unusual, like a double horizontal bar. That means you have mating between blood relatives, incest, gross. And then we also have identical twins and non-identical twins are a little different than the sibling brackets, so just be aware of that. So then you're going to take a look at these case histories. So each case history has some sort of inherited trait. Okay, so it talks about the trait, and then it talks about the family. Okay, so who has it, who doesn't. And then once you've kind of read through the case history, then you're going to draw a pedigree using the knowledge that was given. Okay, so remember, you would set up the circles and the squares, lines, horizontal lines indicating a mating, and then the vertical line indicating the child that was produced, and so forth. And then draw that. Okay. Once you've drawn the pedigree for case 1, then you can use the pedigree to kind of do some predictions of what you think the genetic disease, if it's autosomal dominant, is it autosomal recessive, is it sex-linked dominant, is it sex-linked recessive, that kind of thing. And then do some ethical decisions. So if you were counseling the couple, what are some of the options that you think they would have? Should they try having a child? with an increased risk? Should they maybe choose adoption instead, if it's something that, you know, maybe could cause a lot of harm to the developing child? Could they do something like IVF? You know, so you can think of some scenarios that you could use to advise the parents. Okay, and then you'll also give your own opinion of, if this happened to you, if you were faced with the same situation, what do you think you would do? Okay. And then case history 2 is the same, you know, kind of thing, where you're going to read the scenario and then create the pedigree and then answer the questions. And then finally we have case history number 3, and the same thing, where you're going to create your pedigree and answer the questions. Okay. And then finally on page 289, we have our summary. So you're going to answer the questions, summarizing, you know, what you have learned about blood typing. And then we have some case studies. So you're going to be drawing some more pedigrees to determine like paternity of the children. And also we have a Charlie Chapman paternity suit, so you're going to draw a pedigree, kind of determine that as well. Okay, so once you have answered all those questions, then you'll choose the ones that apply to your student number and post those. Okay, and that is it for this week. Those are the only 4 assignments. We do have some information about our final lab report, which is present here. So now that we've learned about the bitter taste gene, we've done the PTC test, you've learned how DNA gets isolated and run through PCR, and then finally the gel electrophoresis will determine if our student population, you know, how many students actually have that are homozygous dominant, heterozygous, and homozygous recessive for this particular gene. Those results are here. Okay, so obviously you're not going to be, you know, we don't have all of these available for our particular class because we didn't run the PCR for our particular DNA, but these are the results that you're going to be including. So for now, what I would do is on the back of your lab manual, starting on page, where is it? Starting on page 326. On page 326, we have table 1, table 2, and table 3. So table 1, you can go ahead and import this data. So we have 14 students that are non-tasters, 33 that are weak tasters, and 11 that are strong tasters, and then figure out the percentage. And then use what you learned from the Hardy-Weinberg equilibrium, which we did way back in topic 6, step 2, to figure out, you know, what's the predicted PTC paper tasters in the class. Fill out table number 2, and then table number 3 is our PCR results. So, you know, single large band, 13 students, small band, 10, and then double band, 34. And what does this mean? If you have 13 students that have single large band, what are their genotypes? Are they homozygous dominant, heterozygous, or homozygous recessive? So you can enter that information there on page 326, just so that it's available when you go to write your lab report. So this lab report is not due until March 15th. But, you know, you can at least kind of start working on it this week if you want. Okay. And that is about it for this week. You will need to think about your topic 7 quiz. So the topic 7 quiz covers all of topic 7, including last week's material. And that's not due until the following Wednesday. Okay, so you have plenty of time, but, you know, feel free to go back into your Digia textbook readings and continue reading so that you can have some background information on our topics this week, as well as, you know, prepare for topic 7. In the meantime, if you have any questions, feel free to email me. Otherwise, good luck.